Hi there, and I believe you are returning from the previous video on how we began looking at the top hits or top drugs that could inhibit some of the proteins expressed by your upregulated or downregulated genes. So in this video, we will look a little bit much more in depth into how to organize this data and to do other more detailed analysis. So let me also show you one of the cool things you can do with the dataset that you've already retrieved. Remember, you want top target drugs or top hits because, you don't want to spend a lot of money and time in testing drugs that may not work. So you can query back the library to give you only the top drugs based on their adjusted p-value. So to do this, we can simply just copy paste this here and then have a variable called top drugs. And so this is our variable that contains our top list of drugs. But then we can query now this variable to give us the top drugs based on their adjusted p-value, which basically means we can order these drugs based on their adjusted p-value. So we simply need to type in top drugs over there. And then query the top drugs variable that we created earlier. And then use the square brackets and then just use the order command. And just order the top drugs. There's a variable within the top drugs data set called the adjusted p-value that we can use. And tell the program to return for us everything, as simple as that, and then just run that. And then we can now use head to access the top drugs and just return the first 5 top drugs. And you can see that we have the radicizol as the top drug here, and you can see the genes that it targets here. Now, we can make this a little bit much more cleaner in a way that you can send to your PIs or even your supervisors or even your collaborators. So we can create a function that helps us simplify everything, and this will help us avoid any repetitions that may be redundant. We can call the function anything we want, for example, clean enrich R, and then just call the function from R, and then say that the function accepts something called enrich R data frame, and then it also accepts a label. And then we say we want the top n drugs. For example, we can say top n drugs of, let's say, 15 drugs. And then we can write what the function does. So the function basically takes the enrich R data frame and then processes it using the pipe. It converts it to a table. And then once we've converted it into a table, we can select term. We can also select overlap and then select adjusted p-value. And then the library also have something called combined score that we can access. And then we have the genes that those drugs actually target, and then we can process this. We can rename things, rename them with paste. Just see the label that we provided earlier. Separate it by that, and then just a full stop there. That works fine. And then also combine it with the term. So we can simply pipe it back to arrange. So what we want to arrange is label, and then adjusted p-value, and then pipe it to slice head. We want only the top number of drugs that we wanted earlier, top n. And that's it. So we can simply see where the error is. The error is here. We can not type in the best command as that. And that's it. So I will include this function into the GitHub repo. You don't need to understand what this function does, but it basically rearranges all this messy data into something that is clearly readable and something that you can even send to other collaborators. So let's run this. There you go. And then we can simply apply this function. So I can just create a simple variable called top up drugs. There you go. And then run my function clean enricher. And then I want to run it on this data frame here that have got top hits that repress my up-regulated genes. So I just copy paste that here. And then say that the label I want is up. And simply run this command. And then you can now see the list of genes in a much more cleaner way. So we can go to my global environment and look for this variable called top up drugs. It's here. And then you can see that I have ubiquinol as my top drug here, and it targets these genes here, the NF-kappa B1, the Jun force, as well as the HIF1. Remember that here I ran this function on this library here called the drug perturbations from the gene expression omnibus. But I can change this to have from my link library. For example, just do that, top up drugs, and then just rename this to links, and then change this name to the links library. For example, I can just copy paste this here. As simple as that, and then just run it. And then I can just go to my global environment and click on it again, and then I will see that radicizol is my top hit, and these are the genes that it targets. So you can either work with radicizol as obtained from the links library, or you can even work with ubiquinol that is obtained from the gene expression omnibus. Either way, you want drugs that have much more chances of working in your system. So we can also do the same for the top hits that can restore or rescue our down-regulated genes. So to do this, we can simply copy this and paste it over there, and they just rename things to top-down drugs, and then change here to down enrich. Now, because it's down enrich, we want to query it against the up library. So change here to up, and then change here to the label to be down. And you can simply run that. And you can go to your environment and now have a look at those drugs. So you can look at them here. For example, we have the BES235 that targets the CDK1, the GAD45. So there is a whole list of drugs here that you can test within your cell lines. So now with both results, you can now go into further steps to look into literature, how now these reversals sometimes happen, and then proceed to test them within your cell lines. One more cool thing we can do is to have a ggplot of the hits that we have. For example, I can work with the down enrich data frame. I can just copy that and have a variable here called top down, and then paste that over there. And then just query it to have the first up to the 10th hit, and then just return everything that is there. Simply run that, and now we can have a ggplot. So we want top down, and then we can set the aesthetics. So we want X to be where we reorder everything, but we reorder with the term, and then combined score, and then just set the y-axis, just the combined score, and then add the graph that we want, for example, the GM call. So you want the fill to be, let's say tomato. You can change this with whatever fill you want. And then just fill in the coordinates. We can set the labs, have the title, put the title anything you want, for example, top drugs. That upregulate, something like downregulated genes. And then set your x-axis label, drug. We can also set your y-axis label to combine score. And then add something like a theme to minimal. You can set the theme anything you want. But remember, before running this, remember to ensure that the ggplot is activated in your system. In my case, I don't have it activated, so I'll just activate it here. And then run this in case it doesn't have any errors. And then have something like that. Have your top drugs that upregulate your downregulated genes, and then you can have your top drugs as so. So this is pretty much simple. You can show this in your meetings, you can send this to your collaborators, and do any other thing you want to do with it. Otherwise, I hope you enjoyed this tutorial. If in case anything was not clear, you can simply tell me within the comment section, or even post it in my GitHub repository. I will read and respond in time. See you in the next video.